Hey guys, so we're going to go through the last half of chapter 13 in this video. We're going to talk about using chemicals and then also some tests to make sure that what you're using to disinfect actually works and is doing its job. So let's talk about chemicals. There are lots of different ones. The very first one was carbolic acid, and it was used by Leicester. We've heard about him again, right? We talked about how he told his fellow doctors to clean the tools, clean your surgical instruments, and he was using carbolic acid phenol to clean his surgical instruments and clean the wounds. So phenols are coming from Leicester, and Leicester inspired another doctor after that, another scientist. He was actually a chemist, not a doctor, and it was Joseph Lawrence, and he came up with a chemical. It was alcohol-based oral antiseptic, and it was called Listerine. So Lawrence, the chemist, named his chemical that he came up with after Joseph Leicester, who was telling everybody to clean your tools, clean the wounds for surgery. That made a huge, huge impact. And so he named this chemical Listerine after it. And we are still using Listerine today. How do the phenols work? They are going to control growth by denaturing proteins, because proteins are important, their shape matters, and if we denature them, they don't work. If we disrupt the plasma membrane, cells don't make it either. So two different ways that these phenolics can work. So Listerine is one. Lysol, I was telling y'all about, that we can spray on different surfaces. That's a phenol. Hex is another one, and triclosan. All of these are different types of phenols in this category of phenolics. Triclosan is something that you used to see a lot of in soaps to make it antibiotic. Then it was in our water systems. It was everywhere, and pushback came back that we are possibly making superbugs because we're putting these antibiotic soaps out there. Is there a difference? Are we seeing a significant difference when we have triclosan in the soap versus just regular soap? Prove it to me. Let me know that you are seeing a difference. And I want to say the textbook mentioned that, FDA gave soap manufacturers until 2016 to prove that the soaps made a significant benefit over traditional soaps. And if they couldn't, they needed to pull the triclosan label off of their products. So, despite evidence that it poses environmental and health risks, no significant health benefit compared to conventional soaps. And I gotta say, when I first heard this, I was like, no, give me the triclosan and the antibiotics. But now that I know better, and I'm just degerming my hands anyway, and I'm not going into surgery, just regular soap is fine for that. Other things that are chemically based, heavy metals, there are lots of them. The first one that they have up here is copper. Copper is going to, all of these guys are going to kill microbes. They're going to mess with their proteins, mess with their enzymatic activities. So basically, DNA, RNA, proteins, you need the proteins to do all sorts of things in the cell itself. And if you mess with the proteins, it's going to kill the guys. So copper is one that will also, we have copper, zinc, silver, all sorts of different heavy metals out here. Some of these guys will bioaccumulate. It mentions, here it is, Geria, this guy right here. So there's some solution out there that you can get, and it's called colloidal silver. And I personally use a little bit of it when I neti, because when I get sinus infections, it seems to help. But you just use a little bit of it, because if you use too much of this stuff, and it is antimicrobial, it's a heavy metal, colloidal silver, it builds up in your system, and you could end up like this person. And I am old, and I still remember when Oprah had a talk show, like a daily talk show. And I distinctly remember this man coming onto her show, and his nickname was Papa Smurf. because he did this. He took colloidal silver like every day, like I think he took a spoonful of it every single day, and it accumulated in his system and it turned him blue. So heavy metals, they are antimicrobial. They can accumulate. Some of them have toxic effects, so you need to be careful. Mercury is one of those. It is toxic to the central nervous system. But we used it for a number of years. I still remember, because again, I'm 40. So I remember this. My grandmother used to have this. It was in her medicine cabinet, mercurochrome, and you would put that on your scrapes instead of way before Neosporin. There was mercurochrome. If you get a filling today, you probably get some sort of porcelain filling in your mouth for dental cavities, for dental caries. But before that, there was the metal amalgams, and half of the metal amalgam was mercury. It was about 50% mercury. And I want to say they still use amalgams today. And so the evidence is, I think, inconclusive as far as whether or not that is potentially harmful to have those in your mouth, those amalgam fillings, versus the newer, more aesthetically pleasing anyway, white fillings that they use. Silver, if you have copper pots that it's got antimicrobial, silver is also antimicrobial. So water pitchers could be lined with silver, your utensils that you eat with. Silver, antimicrobial properties. Zinc, nickel, there's our handy dandy Listerine. All of these heavy metals can have some antimicrobial properties. Zinc you'll find in mouthwash. Yeah, zinc chloride is going to be in our mouthwash in the Listerine that we were just talking about. So heavy metals, moving on. Halogens, however you want to pronounce it. It's going to be iodine, chlorine, and fluorine. All three of these are halogens, and they are going to, did I put it in here? I didn't put it in here, what it does. But they are going to destabilize the macros, your amino acids, your fatty acids, your lipids, your nucleotides. These guys are going to break those apart, and you need all the macros for your cell. So these are very good at what they do. Iodine, if anybody is going to have a surgery, usually you get wiped down, not usually, but sometimes you will get wiped down with the betadine, the brown iodine, and it's going to disinfect that area. Chlorine bleach is going to be used as a disinfectant. The gas is not good for you. Chlorine gas, they will use chlorine to disinfect our water. Our public water is disinfected with chlorine. If you go swimming in a pool, sometimes it's salt water, but back in the day, they were all chlorine, and you would put a chlorine tablet. That was my first job as a lifeguard out of high school, and you had these chlorine tablets that you would put into the giant pumps to chlorinate the pool. And one day our manager for the pool poured just straight buckets of bleach into a bucket, and we had to scrub the side of the pool with that. And we were all inhaling chlorine gas, and every single lifeguard that day was coughing up a lung because chlorine gas is not good for you. You don't want to inhale that stuff. You need to get away from the bleach fumes. If it starts to accumulate, just air out the room. You don't want to get exposed to chlorine gas. It's not fun. Gonna move on. Oh, fluoride. I left fluoride out. I don't have it in here. Do I have it in here? No, I don't. But fluoride is another one of those. If you go to the dentist, speaking of the dentist and cavities, something to help possibly prevent cavities is getting fluoride treatment on your teeth. And fluoride is one of these too. So it can help be incorporated into your teeth enamel, and it is going to be bacteriostatic. So hopefully it can slow the growth of bacteria around your teeth that are trying to eat into that enamel. It promotes remineralization of the teeth too. Alcohols, if you are trying to find hand sanitizer, good luck. Maybe you can still find some. but they are going to denature the proteins just like all of our other guides before. Denature the proteins. It's going to stop their metabolism. And they can also disrupt membranes, and you don't want the cell membrane, it's going to rupture and lead to lysis. These can be bactericidal, fungicidal, so kills bacteria, kills fungi. They may also kill viruses, but only the enveloped viruses. So COVID-19 just happens to be a virus with an envelope around it. And so these hand sanitizers can hopefully work for you if you get the chance to use them. Otherwise, degerm with soap and water. Wipe it down. And if you have a surface, use a bleach solution if you can't find something else like Lysol, one of those ready-made products. Isopropyl alcohol, rubbing alcohol. Moving on. Speaking of soaps, the surfactants. So what those guys do, they are going to break up the membranes. Surfactants are going to lower the surface tension and bust up those cell membranes. They're going to carry away. When you foam up and wash up, they will wash away the substance. If it's a virus like our enveloped one, we're going to bust up the lipids, the grease layer, with the surfactants. So they usually don't kill unless you've got antiseptics or disinfectants in there, but sometimes the surfactant action itself is enough to get rid of the thing and hopefully get rid of whatever it was before you touch your face and eat your food and stuff like that. Another soap, another group in here, yes, the quats, quaternary ammonium salts. These guys, they have these little rings at the end, and they are going to, you know, our phospholipid bilayer, the heads are hydrophilic and they love water, and so they're on the outside, and the tails are hydrophobic and they're tucked on the inside. They don't like the water. These things, the way these are structured, the way quats are structured, they will bust up the membranes of the cells. So they're able to, there you go, infiltrate the plasma membranes and disrupt their integrity and the cell lysis. So these are quats. Once again, the proper hand washing technique. I did go back to the previous video and add in those little songs that you can sing to yourself while you are performing all of these steps during at least 20 seconds to wash your hands with the soap. Any soap will do. And have clean hands and degerm so you're not going to get yourself or your loved ones sick. This, I can never pronounce this one, bisbiguanines, maybe, I think I got it. It's chlorhexidine. It's chlorhexidine. And so this one goes, it's a broad spectrum. I don't have it in here exactly specifically what it does. The textbook says it's going to go after yeasts, gram positives, gram negatives, and gram positives, with the exception of the Pseudomonas, that guy that likes to grow in immunocompromised people. We talk about grows in the lungs and grows in different places on people that might be more susceptible. I want to say it was a hot tub case too of a Pseudomonas causing that rash. But these guys are going to disrupt cell membranes again, and in high concentration, it causes the cytoplasm to congeal. That just sounds lovely. So this is chlorhexidine. You can use this as a spray. You can use this as a scrub, a surgical scrub instead of normal soap. You can use chlorhexidine and scrub up before a procedure. Sometimes they will use this as a mouthwash if you've gotten, I got bad teeth, y'all. I just got bad genes. I had bad teeth, so I always have something going on in there. And the last time I had a filling done, it was a mouth rinse of chlorhexidine, so it can get in there and make sure that if anything got in there with bacteria-wise, that the chlorhexidine would kill it. Some more of these alkylating agents. These are going to kill lots of stuff. They are going to replace their hydrogen with the alkyl group, and that's how we get into this whole pile. Some of these look familiar. Hopefully we've heard of formaldehyde at least and know that that is a preservative that we use, we'll call it formalin, to preserve animals and people. At least for a little while, formalin will do that. And it is going to be a disinfectant. It's going to kill bacteria. It's going to kill fungi. It's going to kill viruses. And I think let's check the textbook. Endospores too, yes. So it gets all those. It's bacteria, they're endospores. Fungi and viruses can be disinfected with this and sterilized at lower temperatures than heat. So a combo of this stuff and heat would be, let's get through that stuff. I think this might be our last one in this section. Yeah. So our last one is going to be the peroxygens, and we mentioned that when we were talking about the hyperbaric chamber, that we can get oxygen into the tissues, and it's great because the tissues need the oxygen, and the organisms sometimes can't stand those products, those radicals that come from that. So peroxygen is going to undergo the reaction that takes, we've got peroxide that's going to break down into our oxygen molecules and our water molecules, and we can use that to clean wounds. Sterilize. So that's the last of this section. Let's move on to 13.4, and how do we test to make sure that these things are still doing what we hope that they're doing, that they are disinfecting. So one of the things that you can do is called the dis diffusion test. It is something that we used to, if we were doing face-to-face, we'd be able to do this test. So what you do is you take your bacteria in your plate and you're gonna lawn it and cover it all up with whatever bacteria you got. Usually we do like E. coli and B. sub and maybe one other bacteria, and we'll lawn the plate, and then you're going to have these little discs, and they're in this kind of, it's almost like a stamper. And each stamper has a different, each little disc in it is a different antibiotic. And so you can test multiple antibiotics all at once on your plate of E. coli, and you stamp it down, and you're going to see how E. coli reacts to all of those different discs that are soaked up in antibiotic, treated with antibiotic. And you'll see, do the E. coli react, or the B. sub, or whatever it is? Does the organism change its growth pattern? Is it still lawned all over the place and just growing up around the disc? Is it freaking out about the disc and growing away because the antibiotic has killed off those bacteria that are touching the disc? And if it does, we call that the zone of inhibition. So you can measure that, because like, see here, what do we have? We got Pseudomonas. We're just talking about that guy. We have Pseudomonas growing in this plate. And Pseudomonas doesn't care about this antibiotic, whatever that is. It's laughing at that antibiotic, and it doesn't care about this one either. But this antibiotic, whatever this one is here, that one actually works. It has a zone of inhibition, and you can measure it, and we measure it in millimeters, how far out it went, or we can even, that one's even almost a centimeter across. So we will measure that zone of inhibition. Did it work? Did it not work? How big is the zone? Because this zone on Pseudomonas, this one's even wider. Look how much wider this from here to here. And this is Staph aureus on this one. So Staph aureus is susceptible to 1, 2, 3, 4 different antibiotics, whatever those antibiotics are in those discs. There are 4 different antibiotics, and all 4 of them worked against Staph aureus, but only one antibiotic worked against the Pseudomonas. So you can measure it. It's called the zone of inhibition. What else is on this slide? Phenol coefficient is what they started with. Remember, we said one of the first chemicals to disinfect was phenols, carbolic acid from Leicester. So everything at a while ago, we used to compare it, and it was called the phenol coefficient. Does it work as good as phenol, or less than phenol, or more than phenol? If it was the same, it was 1. If it was less than, it didn't work as good as phenol, it was less than 1. And if it worked better, killed more microbes than phenol did, then it was more than 1. But we don't use that. anymore, but it did mention it in the textbook, so I've got it in there. The last test is the in-use test. And so you've got your detergent, your disinfectant, and we've got our bleach water solution. And maybe I put too much water in there. Maybe it's been diluted down and I don't have the right ratio of bleach to water. Is it still disinfecting? I don't know. So we can test that. So it's the in-use test, and we are going to do a serial dilution. We're going to pull out 1 ml of our disinfectant. We're going to put it in 9 ml of broth. So we're going to get our nutrient media, 9 ml of broth, and 1 ml of our disinfectant. And we are going to, and it's got disinfectant in activator. We are going to put it together. We will pull out, we'll pipe that out, some of this 10 mils of solution, and then we're going to put 10 drops in 2 different plates, 10 drops a piece. So we got 10 drops in here, and we put 10 drops in here, and then this one's going to get incubated at body temp, and this one's going to get incubated at room temp. This one's 3 days, this one's for 7 days. How many drops are going to grow? Do you see colonies at this point? If you have 5 or more colonies, then your disinfectant is not disinfecting like it should. So this is called the in-use test, where you're kind of performing a serial dilution using bacteria media and then growing it to see how much colony, how many is it growing? Is it growing a lot of colonies, or is it doing its job and keeping those colonies under control? The last part of this chapter is talking about Roberta. And remember, she had gallstones. She went in for surgery. They removed the gallstones. She came home, and then she started getting real sick. And so her symptoms are worsening. She is got pyelonephritis. Kidney is not happy. So now she's in the ICU. She's continuing to deteriorate. Now she's septic. This is legit serious. So she's septic. They're going to culture her urine and see what's in there. What is the bacteria that's taken over poor Roberta, and we got to figure out what it is. So we're going to culture it and run it through a drug sensitivity test to see, kind of like we saw where we lay on the plate, and you're going to expose it to all the different drugs and see, do all of them work? Does only one of them work? What do we have here? So we're going to expose it to a lot of antibiotics, including carbapenems, and see what happens. And her infection, Roberta's infection, was caused by a superbug, and it is resistant to that carbapenem. So it's an enterobacteriaceae. It's a gut bacteria that's resistant to that drug. So it's called CRE resistant. So she is a superbug. And it's got a 40 to 50% fatality rate. That is not what you want to hear. So she's got the kidney infection. She's in septic shock. She needs help. She is getting treated with dialysis, IV fluids, trying to maintain her blood pressure and prevent her blood from clotting. IV, antibiotic. And then she pulls through, Roberta pulls through. So she recovered from her CRE infection. But then they're like, hold up, somebody else has got the same thing going on. So several patients have started to develop CRE infections. And you know what? They're all at the same hospital, and they all have the same procedure. It's got to be something there. So it turns out that it was the instrument, it was the duodenoscope there. And so the textbook says it in, where was it? Ronald Reagan UCLA Medical Center in 2015 had 179 patients that were potentially exposed by an endoscope that could not be sterilized. It was harboring this bacteria, this CRB, this superbug, in these tiny little places that are really hard to clean. These little crevices were containing this superbug. And so they had possibly potentially exposed almost 180 patients to this contaminated endoscope. It said 7 of the people that were exposed got the infection and 2 of them died. So this is serious business. How are you supposed to clean the scope? Whose fault is it? Is it the scope manufacturer? And they're throwing their hands up saying, no, it's not us. We didn't know. The hospital saying, we didn't know. Who is it? The hospital? Is it the doctor that performed this procedure? Who's responsible? So there's a whole debate about this in there, trying to figure out ways to alleviate it. They changed protocol in the hospital to try and figure out if that might help. If we used a different procedure to sterilize this equipment, will that help? And it was still, CRE was still getting in the equipment to the patients. So it was really interesting to read about this story about this superbug and trying to figure out a way to treat it and figure out where does the problem finally end? How can we fix this? So that's the end of chapter 13. Let me know if you got any questions.